Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Vedika Maraj. This lesson is brought to you by the Gauteng Department of Education in collaboration with Cybono Discovery Center. Today's lesson is on evolution in present times and evidence of common ancestors for living hominids. And it's going to be presented by myself, Vedika Maraj, and Fauzi Rahman. Right, let's get right into it. Firstly, as always, we start with the examination guideline. This gives you an indication of how much detail you need to study and exactly what topics you need to study within your big topic. You can also look at your examination guideline because it will give you the which topics, which paper the topics fall under, as well as the mark allocation for that particular topic. For example, evolution is in paper 2, so you will be able to find it, all that information in your examination guideline. Right, so this is the first part, evolution in present times. Any one example of natural selection and evolution in present times. So I'm going to go through a few of these, but these are the four that you need to know at least one example of these, right? So we're going to go through a few of them. So as you know, evolution happens gradually over many, many years, okay, over millions of years actually. But we do have some examples of evolution happening in present time. And this is because we have rapidly reproducing species. And these allow us to study evolution in a short period of time. So let's look at some examples of this. Let me first talk to you about HIV, right? So the HIV virus, there's no cure for it, but there are drugs that you can take in order to assist the symptoms and in order to assist the disease. And these are called antiretrovirals, okay, or also known as ARVs. Maybe you know them as that. So what happens is that some people don't take their medication on time, okay, or they forget to take them. And what happens is that this virus starts mutating rapidly, and it starts mutating, and it comes up with different variants, and it becomes stronger. And the ARVs, when you start taking them again, they're not going to be working as effectively. Okay. Another example we could look at is the coronavirus. Now, we don't know much about this yet, but what we do know is that it was also mutating rapidly. And that's how we landed up with all these different variants, like the Delta variant and the Omicron. Okay, so we are still basically living it, and it's changing and mutating all the time. So we don't really know what the outcome is going to be. Right. With regards to the Anopheles mosquito, that's this one over here. This mosquito is a malaria-carrying mosquito, right? So there's been studies done where they have actually treated crops with DDT. Okay, DDT is an insecticide. So what they did was they treat the crops with the DDT to kill the mosquitoes, right? And the one type of the mosquito is the Anopheles mosquito. So they would spray the crops, and then those mosquitoes would die because they are the pests, right? But over the years, they realized that because of random mutations, a part of the population actually became resistant to the DDT. Okay, which means that when the DDT was sprayed, those ones didn't die. The ones that were resistant, they did not die. So they passed on, they passed down that gene for being resistant to DDT down to the next generation, okay, or to the offspring. And then over time, it eventually you'd get a population that is more DDT resistant. Okay, so and also DDT is also banned in many countries because it also has terrible side effects. One of the side effects is that it actually kills other organisms and not just the Anopheles mosquito. Right. Now we look at, oh, and this is obviously happening because of natural selection. The process that I just explained about having the gene for the resistance and then passing that on to the offspring, that is all part of natural selection and survival of the fittest, right? So let's look at this one at the bottom when we're talking about resistant bacteria. So example of this is TB. Okay, so you actually get TB that is resistant to TB antibiotics. Okay, and this is an example. This is just a diagrammatic explanation of what I was explaining to you with regards to the DDT and the mosquito. Okay, so let's have a look at it. So if we've got a population of mainly susceptible bacteria, so that means that these bacteria are actually going to be killed by the antibiotic, okay, because they are susceptible. Whereas the resistant bacteria, that means that those are not going to die when the antibiotic is going to be administered, okay? They are resistant bacteria and they are resistant to the antibiotic. It's very important that we understand the difference, okay? The bacteria is the disease causing pathogen, right? And the antibiotic is the medication that we take to kill bacteria. The resistant bacteria are the bacteria that are not killed by the antibiotic because they are resistant to it. Okay. So in this population, because of a random mutation, we have these two resistant bacteria, the ones that are indicated in red, that have now developed in this population. Now we have the administering of the antibiotic, and then you can see that, so what will happen is that all the yellow ones will die out because they are susceptible, right? They are able to be killed by the antibiotic, and what will be left will be the two, or majority of them will be killed, and what will be left here in this picture are the two that are resistant, okay? And then this will carry on going, and over a long period of time, we are going to then land up with this picture here at the end that is showing us a population of mainly resistant bacteria. Okay, so now I've given you the background. Now, how do you answer questions on this? Okay, very important. So we're going to do two activities that's going to just cement what I've explained to you. Right, so we go for the first question. This is taken from the DBE November 2021, paper 2. Some farmers add low doses of antibiotics to the feed for cattle. The use of antibiotics in cattle feed could result in the evolution of antibiotic-resistant bacteria. The graph below shows the effect of different doses of antibiotics on the number of harmful bacteria in the cattle. Okay, so very importantly, I forgot to mention, which you would know that I almost always do, you must always read the stem of your question very carefully. Okay, read it twice. The first time you read it, just try to get a feel of what is this about. The second time you read it, you go into a little bit more detail. You try to ask yourself questions. What is this activity, this information telling me? Okay, look at the graph, analyze the graph. Right, so let's have a look at the graph. Here we have on the x-axis, we have the antibiotic dose, which is measured in milligrams. Very important to also take note of that, because if they have to ask you to read anything off the graph, you're also going to include the unit, okay? Which it's given over here. And then on the y-axis here, we have the number of harmful bacteria. So they're showing you here, if you can look at this, and also let's look at the caption. Let's look at the caption. Effect of different doses of antibiotics on the number of harmful bacteria in livestock. Okay, so they're showing you here at 2 milligrams, at 4, at 6, at 8, and 10, what is happening to the number of harmful bacteria. And as you can see in this diagram, as you are administering a higher dose of antibiotic, the number of harmful bacteria is decreasing. Okay, that's the main information that you're going to get from this. The other thing, yes, the other main the sentence that's important is this one over here. The use of antibiotics in cattle feed could result in the evolution of antibiotic resistant bacteria. Okay, let's have a look at the questions. I just kept the graph there because we can refer to it so you can easily see what it is that we're looking for. Number 1, use evidence from the graph to explain why higher doses of antibiotics will benefit the farmer economically. Evidence from the graph. Explain why higher doses of antibiotics will benefit the farmer economically. Okay, why am I underlying these words? Because it's 4 marks and it's actually quite a loaded sentence. Okay, so we need to break it up for ourselves. What is the examiner wanting me to answer? Okay, firstly, you need to use the graph. When they tell you for evidence from the graph, they want you to use the graph. Then they want you to explain why higher doses of antibiotics. Okay, so why will explain why it will benefit the farmer economically. And when we talk about economically, we are talking about the money. Right. So how is this going to benefit the farmer to make a profit or to be able to make a living, okay, to make more money? So let's think about it. Let's look at the evidence from the graph. So the graph is basically showing us that the higher dose of antibiotic we use, the less the number of harmful bacteria there are. Okay. So that's a good thing, right? Because we don't want the bacteria in the cattle, in the livestock, because they are harmful bacteria. Remember that we also get good bacteria, right? In the gut of our tummies, okay, in our stomachs, especially with also in the livestock, okay. But now these are harmful bacteria that are actually causing them to die or causing them to get diseases. So if we have cattle that has a disease, it's not going to really benefit the farmer much, right? Because they're going to be sick or they're going to die. So how do we answer this question? How do we put in words what we're trying to say? The first thing we indicate is we use the evidence from the graph. What do we see in the graph? And that's the first bullet point. It decreases the number of harmful bacteria the most. Okay? Because it's the higher dose. Here's the higher dose. The highest dose, this is the highest dose. Here we have the highest dose, okay, at 10 milligrams. And then if you read it off the graph, it's the lowest number of bacteria. The second bullet point telling us that this prevents disease in cattle, or you can say it results in less medical expenses, okay? It also decreases mortality, or you can say maintaining the number of cattle, because basically less cattle is going to die, right? So that you have more to sell or breed, or it's going to increase your profit. Okay, so when you're doing an explain question, answering an explain question, you must always think of the cause and the effect. Okay, so what is happening here is that we have a decrease in the number of harmful bacteria. Okay, so therefore, what is that going to cause? It's going to cause less diseases to happen in the cattle. It's going to have less cattle to die, and less cattle will then die, and therefore the farmer will have more cattle to sell or to breed, and he's going to get a profit. Okay. Right, the next question. Explain how the use of antibiotics in animal feed, another explain question, in animal feed may result in the evolution of antibiotic resistant bacteria. So when they're talking about this evolution of antibiotic resistant bacteria, you must already be thinking, what? Natural selection. Okay? That is the word or the term that you need to remember. Right, so let's have a look at it. How do we go about it? You actually go about it the exact same way that you use your genetic template of natural selection, and you apply it to the given scenario, okay, or the question, the example that's given to you in the question. Okay, so let's first start with, they're talking about, okay, well, firstly, you need to mention that natural selection occurs. It doesn't have to be there. You can mention it somewhere else as well. We normally start mainly with this first point to say that there is variation. Okay, there is variation or the mutation in the population of bacteria. So there's a variation or there's a mutation. Okay, those are the words that we use. Some are resistant to antibiotics and some are non-resistant. Okay, so that's the variation in them, or that's the mutation. That some are the resistant ones. Okay, that's usually the ones that mutated, and some are non-resistant. When the antibiotic is added, the bacteria that are non-resistant are killed by the antibiotic. Those that are resistant survive and reproduce. The characteristic for resistance to antibiotics is passed on to the offspring. The next generation will have higher proportion of antibiotic resistant bacteria. Okay, so how do we go about this? Like I said, we first start with, okay, in this instance, they are mentioning natural selection. You can also mention it lower down, but let's start with it the way it does here. Natural selection is going to occur. How is it going to occur? Originally, there is variation in the population. right? Or a mutation has occurred. Then you mention the characteristics. Some are resistant to antibiotics, some are non-resistant. Then you explain what happened. When the antibiotic is added, the bacteria that are non-resistant are killed, okay, because they're able to be killed by the antibiotic. The ones that are resistant, they are the ones that are resistant to the antibiotic. They survive, okay? And when they survive, they reproduce, right? Because that's what bacteria does. The characteristic for resistance then, because those ones are resistant, so that characteristic is going to be passed down to the offspring. Then the next generation will have a higher proportion of antibiotic resistant bacteria. Okay, right, let's look at another example. Right, here we have the DBE February March 2018. Study the extract and the information provided. An insecticide is used by farmers to control insect populations of Plodia interpunctella, which feeds on stored grain. Farmers treat the grain with the insecticide to prevent an insect infestation. The insecticide is extremely poisonous to certain insects, yet causes little or no harm to humans and beneficial insects. In recent years, it has been noticed that this insecticide is no longer effective in controlling insect populations of Plodia interpunctella. Scientists hypothesized that insect populations that had previously been exposed to the insecticide had a higher survival rate when the grain was treated again. In an investigation to test this hypothesis, they identified storage bins that had previously been treated with the insecticide and bins that had never been treated with the insecticide, collected a sample of 300 insects from each bin, kept each sample in separate container of equal size and the same conditions, sprayed the same concentration and volume of insecticide over both containers, allowed 24 hours of the insecticide to take effect, counted the number of insects that survived in each container. Right, so these are the results, right? We have previous exposure. With previous exposure to insecticide, the number of insects that survived was 182, and when they had no previous exposure to the insecticide, it was 66. Right, so let's give the independent and dependent variable. When you've been given a table, most of the time the independent variable is the one that is first, okay, and the dependent the one that is second. But that's not the only way to look at it. You also need to ask yourself, okay, which one is the one that is controlled by the person who's carrying out the investigation, okay? And the one that can be controlled is the previous exposure to the insecticide, okay? And then what is the one that changes because of that one? Okay, which is the one that is changing? And that is the number of insects that survived. Okay, so let's check if we are correct. Yes, it's the independent variable is the exposure to the insecticide, and the dependent variable, the number of insects that survived. Right, another question. State 3 factors that were kept constant in the investigation. When we are looking at constant factors, these are factors that are going to ensure that we have validity, okay? It's going to ensure fairness. Validity means fairness. Okay, so it's going to ensure that our investigation was fair. So which variables have to be kept constant so that this investigation can be valid? Right, so when we look at these, we think of things like the sample size, the size of the containers, the concentration of the insecticide, the volume of the insecticide, and the time period. And all of these must be kept the same. Okay, so we can actually start with same. Okay, so we have sample size, the size of the container. Okay, we cannot have one being a big container and one being a smaller container because that can affect our results. The same with the concentration of the insecticide. If we spray more with the one, how are we going to actually deduce whether they died because of the insecticide or because of another variable? Because we applied different doses. Okay, so we need to apply the same concentration. The volume of the insecticide as well, so how much are we going to give, and then the time period. Give 2 reasons why scientists' results may not be reliable. Okay, so why would this scientist's results not be reliable? So now you flip it. You first ask yourself, what do you know about reliability? Okay, we've been saying it many times in over our lessons, but basically, if you haven't been listening to them, if we have to increase reliability, we're asking ourselves what will make our investigation more reliable? What will make our results reliable? How sure are we that these results are correct, okay, or accurate? And the way we do it is that by increasing the sample size, okay, so we use a bigger sample size. Also, we repeat the investigation, okay, so you do it again to see that you got the same or similar result. So now you flip it, okay, and you ask yourself now, if those are the two things that ensure reliability, why would these scientists' results not be reliable? Okay, so you go back to the question and you ask yourself, what did they use? So let's look at the sample size first. If you would remember, they used 300 insects. Now, if you look at the insect population, that is not really a large sample size. Okay, so we can say that they didn't use a large sample size. Okay, so yes, that one is there. They used a small sample. And then another one is that they said that the investigation was only carried out once. So that is also decreasing reliability, because if you want your investigation to be reliable, you should repeat the investigation. Okay, or you can say they did not repeat it. And then another one is that they only used 2 storage bins. Okay, so it's so similar to the sample size answer. State a conclusion for this investigation. Now, when it comes to conclusions, you go back to what was the investigation, what was the aim of the investigation, or if they gave you the hypothesis, you're basically answering the hypothesis. Okay, and in this instance, we could clearly see that the insects that were previously exposed to the insecticide had a higher survival rate, and insects that were, or you can say insects that were not previously exposed to the insecticide had a lower survival rate. Okay, that will be your conclusion. So you look at the results that were given and you come up with your conclusion. Okay, so you have your two variables, the independent and the dependent, and what happened, what was the relationship between the two. Okay, right, now we're moving on to the next topic. And this is evidence of common ancestors for living hominids, including humans. So here we're going to interpret the phylogenetic tree. We're going to show the place of the family Hominidae in the animal kingdom. Then we're going to do characteristics that humans share with African apes and also the anatomical differences. Okay. Right, so our place in the animal kingdom. So some of these you will actually recognize from grade 10. Okay. Also in grade 11, where we have these different domains and kingdom, the animal kingdom. Okay, so where's our place? So we are here, the Homo sapiens. Our species are here at the bottom. We are the Homo sapiens, right? Our genus is Homo, right? This is this one. Our family is Hominidae. Our order are the primates. Class, Mammalia. Phylum, Chordata. And we under the kingdom Animalia, and the big domain, we are Eukarya. Okay, so this is where we fit in to this whole process. I've got another slide that shows you a little bit more detail with regards to evolution and where do we actually fit in the animal kingdom. So we're looking here mainly at this highlighted one in purple, family. Okay, our family name is Hominidae. And then we have our subfamily, which is the Homininae. So if we look here at the Homininae, lots of ninanes, okay? But just look at it carefully so that you can see the spelling to see the difference between them. Right, so here we have the main thing that I want to actually bring to your attention here is that when we are looking at human evolution, we are looking at this portion, okay? Human evolution, actually, it's this one over here, which we go to the hominy, and then we got the Homo. Then you can see at some point we have a branch off with the chimpanzee, okay? And higher up, the first branching we actually have is the greater the apes over here. We have the gorilla, okay? And on this side, on this branching, this is what gave rise to our Homo, and then the pan, okay? But another reason why I have the slide over here is just to mention to you how scientific names are written. So also it started in grade 10, okay, where you actually learned how to do these. Now, if you've noticed, when we look at scientific names, when it's typed out, you will notice that they are in italics, okay, which means they're written a bit skew. But when you write and when I write, we can't write in italics, okay? So how do we indicate the difference with scientific names? And this is how, okay? This is the scientific name, Homo sapiens. So the first word is the genus. It's always spelled with a capital letter, okay, and then the word, and then the species name, the second one, is with a small letter. The first letter of the second word is a small letter, and this whole sentence must be the whole both these words, sorry, the whole scientific name must be underlined. Okay, so we can't write in italics, so it must be underlined. Okay, right. So like for example, gorilla, pan troglodytes, and they can also be shortened as follows: H dot sapiens, okay, capital H dot sapiens, and once again you will underline it, okay, especially when we get to human evolution where we have these huge words, Australopithecus, Ardipithecus. You can just say A. If it's Australopithecus africanus, you can say A dot africanus, but Fozy will go into more detail on those. Then just to bring to your attention that genera is the plural of genus. Okay, so we have one genus and many genera. Okay, right. So then we have phylogenetic trees. How do we interpret this phylogenetic tree? But before we get into that, I just want to show you more or less where do we fit in this tree. So we're showing you on the human evolutionary path, right? Where do we actually fit? So if we look at this particular tree, it's showing us here the past, and we're coming up to today, which is the present day. And we are here at the top, right? You are here because we are still existing. So we are here at the present day. Coming down to the bottom of the stem, when we are talking about common ancestors, we are at this dot over here. Okay? So this is the common ancestor. So what we are saying is that this common ancestor is what has given rise to all the other that are coming after it. Okay? And these that are coming, branching off, we say they're branching off, we're saying speciation has occurred when they branch off, and this branching off, we have the common ancestor, and these that are branching off are the descendants. Okay, that's what I wanted to explain. That we have a common ancestor, and then we have this branching off, and those are the descendants of the common ancestor. Okay, it's important that we get these words correct. So here we have the first branching off, we have the Ardipithecus group. You can see that they fit on this part of our timeline. Okay, so as we are moving on, then evolution is occurring, these species are evolving, speciation is occurring, and we have them developing into new species. Okay, so here we have the Ardipithecus group. So they develop certain characteristics to be fitting into that group. And then we have another branch over here, the Australopithecus group, where they have now gained other characteristics. Okay, and then we're moving up to we have another branch here, the Paranthropus group. And then we had another branch, which is the Homo group. Okay, and we fit into the Homo group. Okay, right. Let's go into phylogenetic trees into more detail. First, let's see, what is a phylogenetic tree? A phylogenetic tree is a schematic form that shows the evolutionary relationships within a set of organisms or group of organisms. Right? If we break up the word, philo it refers to the organism's phylum group, and genetic is from the genes or relationships between the genes. Okay, you should not compare this phylogenetic tree with pedigree diagrams. Okay, don't confuse them with pedigree diagrams, which is the genetic relationship between close family members. Okay, so the pedigree diagram is basically like a family tree. Okay, it's showing you the relationship between close family members. You studied that in genetics. Okay, but the phylogenetic tree is showing you the evolutionary relationships within a set of organisms or between different species. Okay, it's showing you the evolutionary relationship between them, not necessarily like close family members, as in like, husband and wife and cousins and those people like in your immediate and extended families. Okay. Who is the common ancestor? okay, these are the things that we need to think about, right? When we are looking at phylogenetic trees, we ask ourselves, who is the common ancestor? Who is extinct? When did the species on the diagram originate? How long did they live for? And when did they become extinct? Okay, those are the kind of questions that we ask ourselves. Those are the kinds of questions that you are going to be asked in the examination when you get faced with the phylogenetic tree. Okay, and phylogenetic tree, they're awesome. I love them because you know what? It's not even study work. If you can understand how to answer a phylogenetic tree, if you can get the skill behind it, right, and you listen very carefully to how I'm going to explain it to you, and you practice, you can actually answer any phylogenetic tree on any information that is given, because it doesn't matter whether we are telling you about the species of elephant or the species of a rhino or a bacteria, whatever it is. But if you can just read the phylogenetic tree, you'll be able to answer the questions. Okay, so let's get into it. This is showing phylogenetic tree may also be represented in this way. Okay, so you saw that similar to the one that you've actually seen with that big tree. Okay, in this phylogenetic tree, it's important that we first ask ourselves, what is happening here? Okay, so if there isn't a graph given, in this instance, you're going to see this. There's information on the left hand in the green block. There's information there that's actually giving you information on how to read that off. Okay. So you're just having a look there. Let's read and see what the information is telling us, and then I'll explain to you how the branching is occurring. Okay, so when you look at the phylogenetic tree on the right, you will see lots of branches. Okay, you can see that, right? There are many branches. The first branching or speciation event is at number 1. That is the common ancestor for all the species in this phylogenetic tree. Okay, so this is the common ancestor. That's what we need to first establish. Okay, so it's at the bottom. So when you're reading this phylogenetic tree, you're going to read it from the common ancestor being at the bottom and giving rise to all the species, going up. So what they're indicating to us is that this common ancestor is the common ancestor for A, B, C, D, E, F, G, and H. Okay, all of those species. A, B, and C have a common ancestor at number 2. Yes, so what's happening over here is we're going to have 2 branches. Okay, so if you look at this first branch, this one over here, we can see that this descendant going up here, at this point we have another branch. Okay, because now new characteristics have developed. And number 2 is the common ancestor of B, A, B, and C. Okay? But then there was another branch here. There was another common ancestor. This common ancestor number 3. So more developments occurred, okay, or more characteristics changed, and a new species was evolved. So we have a common ancestor here, which gave rise to B and C. Okay, so these are closely related. B and C are closely related. Okay, they are more closely related than they are to A. Okay, and all of these are even more closely related to each other compared to being to this branch. Okay, you see, because the original diverge was here at the bottom. Okay, so let's see what more information they give us. I think we spoke about the second point. The third point, B and C is more closely related to each other than to A. Yes, I mentioned that. And the last bullet point, A, B, and C, that's this one, is more closely related to each other than to D, E, F, G, and H. Okay, we understand that. Right, let's go to another one so that we can see a different type of a phylogenetic tree. Right, in this one, we have, this one is actually, there's 2 things I'm going to discuss with you here. Here I'm going to discuss with you phylogenetic tree, okay, how to read the phylogenetic tree. And then I'm also going to explain to you, show you that this is the history of the hominids. Okay, and Fawzi is going to go into a little bit more detail about she will mention to you these some of these, and then yeah, let's look at it. Let's look at the phylogenetic tree. Okay, so now we have the time at the bottom. Okay, and they are showing you now something funny about this graph. Right, is usually when you're reading a graph and you're reading it like from left to right, you are seeing the numbers 0, 1, 2, 3, 4, 5, right? But in this one, we are showing you, you can see it's going this way. Okay? That we have from, if you read it going this way, we have 5 million years ago on this side, and it's going to the present day. Okay, that's very important. So they are showing you that this is the past and this is the present. So whenever you get a phylogenetic tree, the first thing that you need to establish is when is the past and when is the present. Okay, so let's start here at the past. Okay, and then before we do that, I want to bring this to your attention. Million years ago. Okay, we also write it as MYA. Right. So this is the million years ago. If we're looking at if we're reading off something, this graph, if we're reading off a value from this graph, right, and we're looking at the x-axis, if it's at 3, it is 3 million years ago, right? But if I have to ask you, for how long was a specific organism in existence, right, then you will work out a value, and that value will be measured in million years. Okay, because it existed for 3 million years. Okay, it wasn't that it existed 3 million years ago. We'll do an example after I explain to you how we are going to read this phylogenetic tree. Okay, so we start here with the common ancestor. Okay, this arrow is showing us the common ancestor is at this point, and then we have the first branching off. Okay, branching off into 2. So these are still related to each other, but they are branching off now because they are forming different characteristics. Okay, and speciation is occurring. So these 2 are giving rise to our descendants. Okay, so this is the common ancestor, and these 2 lines are the descendants of the common ancestor. So let's see this bottom one. This bottom branch is the one that gave rise to our chimpanzee. Okay, the upper branch is what gave rise to our Australopithecus afarensis and our hominids that are leading up to our Homo sapiens. Okay, we are over here, the Homo sapiens. So you have this branch, we have Australopithecus afarensis, and then we have another branch at this point, right? So at this point, we have another branching off, which was the A. robustus. And over there, we have another branching off going into the Homo habilis, the Homo erectus, and then we have one more branch over here coming to the Homo sapien. Okay, so all of those dots, where the point, those points of before they diverge, those points are the points where you'll find the common ancestor of whatever the descendants are going to be after that. Okay, so those are the common ancestors. So what else can we read off from the graph? Another thing that we can read off is when they became extinct. Okay, so as you can see, A. robustus, the line just stops there in the middle of our graph. Okay, so if it is stopping there, it means that it is not present now. It's not in the present day. Okay, it stopped there, which means that it became extinct. So if you see a phylogenetic tree and all of a sudden the line, you're following the line and the line stops, you must know that at that point, that species became extinct. Okay? Now let's look at the Homo sapiens. Now you know that we are still here, right? We are Homo sapiens. We are all still here. So you can see that we are going right up into 0, the present day. 0 is the present day, okay? So we're going right up into that line, the present day, which means that we are still in existence. Okay, we are still existing. So the important thing that you must remember about phylogenetic trees is that whenever you ask to read off any value or to work out anything from a phylogenetic tree, you use a ruler. Okay, you use a ruler. How do we do that? Let's look at an example. Okay. When did A. robustus become extinct? What do? You take a ruler, okay? And then you go to A. robustus, you follow A. robustus's line, and you see, he stopped somewhere there. So you take your ruler, you draw your line all the way down. Oh, see, even further. Let's pass the chimpanzee line all the way down to that point. And then you read it off. Now be very careful. Right, which way is your timeline going? Which way is your graph going? So look at 0. Here's 0, which means we're going from this way. 0, 1, 2. Okay? So this value is between 1 and 2. It's the middle. It's about the middle of between 1 and 2, so it's 1.5. 1.5 million years ago. Why? Because it happened in the past, right? Million years ago. That's how many million years ago it became extinct. It is not 2.5, okay? Remember, you must see how the graph is written. Some people say 2.5 because they're saying, okay, normally your graph goes, I explained 0, 1, 2, 3, 4. So now you just make sure your numbers are going in the correct way and you know how to read it off. Another question you get asked is maybe, how long was A. robustus in existence? How long did it exist for? Right? Now we're going to have to read off 2 points. So you take the point of when they appeared, okay? So the point at when they appeared was at this point over here, okay? So let's go. That's the one branching off, right? So we go down. It is around about 3. You must use a ruler, okay? I don't have a ruler here and my line's going a bit squiggly. You must use a ruler. It's at 3 million years, okay? And then you subtract when they became extinct, right? So you take the value of when they appeared and you subtract the value of when it became extinct. So it's 3 minus 1.5, and the answer is 1.5 million years. Why is it million years and not million years ago? Because it was in existence for that period of time. It lived. That's how many years it lived for. Okay, that's the difference between million years and million years ago. Okay, so from this thing, let's just sum up quickly before we go into some examples on phylogenetic trees. Firstly, identify which way to read the graph. Okay, where is the past, where is the present? Then identify your common ancestor, right? Identify your common ancestor, see how it's diverging, and understand that all of these are the descendants of that common ancestor. Okay? Next, everything that the lines that are still extending to present day, those are the species that are still in existence. Any lines that die off or end in the middle of your phylogenetic tree or your graph, that is when that species became extinct. Okay? When did they appear? You trace it back to the branching line. Okay? Or that's when they appeared. That's the first, that branching. And that branch is also, that point before they diverge is also a point of another common ancestor of the next descendants that are coming. Okay? Right. And then million years and million years ago. Don't forget the difference between that. And lastly, use a ruler. Okay, so let's go through an activity. Okay, now this graph or this phylogenetic tree is a little bit different. Okay, so we're going to spend a little bit of time on it so that we can unpack it, because really when you get something, when you get any sort of a picture that you need to analyze, okay, or you need to figure out what's happening, you must unpack the diagram. Okay, you need to unpack the diagram. You need to look, what is this diagram actually telling me? And once you unpack it and you talk about it and you think about it and you write on it the way I was writing, by the time you look at the questions, you will see that you've actually answered most of them. Okay, so let's have a look. First, reading the stem of the question. The diagram below represents the possible evolution of humans, as well as the time period for the evolution of bipedalism, use of stone tools, and use of fire. Right, so they're showing us bipedalism, the use of tools, and the use of fire. Let's look at this graph. First, what did I say? Identify past and present. Here we have the past. It's 5, 4, 3, 2, 1. Here's the present. They even put it as for us there. Oh, sorry. They put it for us there, present. Or it's at 0. Okay. And now they're showing us, let's look at the key first before we go into it. They're showing us the darkly shaded block is Australopithecus species. the horizontal diagonal lines, sorry, the diagonal lines, not horizontal, the diagonal lines are the Homo species, and then the ones with the dots are the Ardipithecus species. Okay, so it's showing us that. Then it's showing us where the bipedalism is, where the use of tools are, and the use of fire. So this block is indicating to us a period. Okay, so this block is showing us that between this time, to write nicely, between this time and this time, that is when fire was used. Okay, and they're showing us between this time and this time, that's when tools were used. And bipedalism was between this time and this time. Okay, then the next thing we need to look at, where are these species, okay, and what are these showing us about that? So let's look at this one that was long time ago, A. ramidus. Okay, so you don't know these words yet, okay, don't stress, right? Just think about the skill that I have been trying to explain to you. Okay, so now we look at this organism, A. ramidus. So this one was more, it's many million years ago. Okay, it's more in the past compared to the other blocks. So we can understand that this one is older. Okay, but let's look. We have A. ramidus. This whole block, you can see here that it appeared just before the 5 mark. Okay, around 5 million years ago. And then it existed for this whole block over here. Okay, so then it became extinct at this point. Why? Because the block ended at that point. Okay, so even this doesn't have to be a line. It appeared, the appearance of it was the beginning of the block, and the extinct was the end of the block. So if you measure that whole block, that is the that's how long it was in existence for. Okay, then we have the other ones. They are following the same sort of thing. I just wanted to explain to you, the beginning of the block is when it appeared, and the end of the block is when it became extinct. Okay, now let's look at the questions. Name the 2 genera that were bipedal but did not use fire. Okay, so now we have these ones used fire, so we cannot look at these ones here. Okay, they said they did not use fire, right? So we need to look somewhere here, but they were also bipedal. Okay, right? So they're saying that they were bipedal, but they did not use fire. The 2 genera, right? It's the Australopithecus and it is the Ardipithecus, right? So how do we know? We go there to when they were bipedal, and we see here that it is this one and this one, right? When did the A. afarensis evolve? So we go to A. afarensis, that's this one over here. And then when did it evolve? We go to the beginning of that block. And it is, we go to the beginning of that block and we use our ruler and we trace all the way down, and it is 4 million years ago. How long did it take for the hominin species to start using fire after they were bipedal? Okay, so once they became bipedal, how long did it take them to use fire? So what we do is we go to the start of the bipedalism, we draw the line all the way down, it comes to that point, which is 4.5. And then you go to the point when they started to use fire. That is at 1. So then we say 4.5 minus 1 is 3.5 million years ago. Okay? Because they asked, how long did it take for the hominin species to start using fire? So when did they become bipedal? You take that amount, subtract by when did they start using fire, the beginning of that, and you get to 3.5 million years ago. Right, let's look at one more example before we go for the break. Right, once again, unpack the diagram. In this one, they are reading us from the past is here at the bottom, and the present is there at the top. Okay, so this one you're reading from the bottom up. Okay. The previous one, you were reading this way. Okay. So let's have a look at this one. They are showing us here, this is the hominin common ancestor. Okay. So this one is the common ancestor of all of these species that are being shown here. Okay. What is this type of diagram called? Yes, very important question. Almost always asked. Okay, what is it called? It's a phylogenetic tree, not a pedigree diagram, a phylogenetic tree. Okay, it's showing you different species here and the relationship between them. How many of each of the following are represented in the diagram? Okay, so how many genera are there? Remember, I told you genera is the plural of genus. So you count how many different genus names do you see. So I see Ardipithecus, that's one. I see Australopithecus, that's two. I see Paranthropus, that's three. I see Homo, okay, that is four, right? And there's also chimpanzee, which is Pan, which is five. Okay, so that's why there are five genera. Then how many Homo species are there? Okay, so there we have Homo habilis, one. Homo erectus, this is Homo habilis, that's one. Homo erectus, that's two. Homo sapiens, that's three. Homo neanderthalensis, which is four. Okay. Then we go to name the species that have Paranthropus aethiopicus as a common ancestor. Okay, so we go to that word, that very long word there, Paranthropus aethiopicus. Let's see, where is it? Here it is. Okay, it's at this point over here. Now they're saying it's the common ancestor of which two species? So like I told you, go there, you find the common ancestor. You see what are the descendants on this line. And we come to Paranthropus bossai and Paranthropus robustus. Okay. Name the hominid species that existed at the same time as the Homo sapien. Okay, so if you look at the Homo sapien line, try to think who was in existence the same time as he was. Remember, so you go to when did the Homo sapien appear? It was around about this point, right? Okay, showing you this branch. So, and it's in existence here. Who was in existence at the same time? It is the Homo neanderthalensis. Okay, because you can see this branch is showing you that if you look at the timeline, they were existent at the same time. Name the first Homo species to appear. The first Homo species to appear, let's go, where's the first one? It is here, it's Homo habilis. It's at this point. We take a ruler, please, not my squiggly line. And we get to a point over there. Oh, sorry, that they just wanted to know who it was. See, read the question very carefully, very important. They just want us to name the first Homo species to appear. That is the Homo habilis, okay, because you can see here from the Australopithecus, we branched off into the Homo, and the first one was the Homo habilis. And if they had asked you when did it appear, then you would take your little ruler, you put a line to the cross, and then you would come here to 1.8 million years ago. Right. That's all from me. We're going to take a 5-minute break and then we will be back.
afternoon, learners, and welcome back. Thank you to Vedika for an awesome presentation. I am going to move on now with the differences and similarities between humans and apes. Now, this section comes out in the exam every year, and learners don't seem to do well in it. So please listen carefully, make sure you study it, and it's easy marks to score. Right, so if we look at the chimpanzee and the gorilla or the apes, there's just some diagrams here for you to see, right? I need you to understand that in order to do well in this subject, you need to understand that you need to keep your religious domain and your scientific domain separately, right? And what we are doing now is we are learning the subject to prepare our learners or to prepare yourselves for the exams. Right, so let's look at the similarities. So we have here the similarities between Homo sapiens and primates. Now, with the similarities, guys, you need to be careful and note what the examiner is asking you. The examiners now are not saying similarities between Homo sapiens and primates. They are saying, let's give me the similarities between Homo sapiens and primates with regards to the head region. Give me with regards to the arms, or with regards to the pelvic area or the pectoral area. So you've got to be very careful when you are writing your exams, and you read the question carefully to find out exactly what is the examiner asking me about. Because the questions, if you go over past papers, you see that the questions are changing every year. So we will ask you the same thing, but maybe in a different way. So if you read the question carefully, you're sure to get this right. Right, let's look at the first part, okay? The first one, if you look at the head region, right, there's large brains and skulls compared to the body mass. So we share with the we Homo sapiens share with our primates large brains compared to our body mass, right? Or skulls. Remember, skull is your top head region, right? Then our eyes in front, two eyes in front, and that gives us binocular or stereoscopic vision. Okay, so that's the eye. And then we're looking at the next one. We're looking at the bottom part now. We are looking at the arms. So there's freely rotating arms. Okay, this is the purple one now, freely rotating arms. And then we're looking at the long upper arms that we have. Okay, this is what we share with them. And then the elbow joints, rotation of the elbow joints, allowing rotation of the forearm. And then now we move to the fingers, right? There's 5 fingers. And an opposable thumb. So this thumb opposes the manner in which the fingers move. The fingers now are moving down this way. The thumb moves this way. So it's opposing the movement of the fingers. It's called an opposable thumb. It works in the opposite direction to the fingers. So that's important for the hand. So if you could see, it's the opposite direction to the hand, as I was saying. Right, let's move on. Now we're looking at the fingers, right? The nails, the flat nails instead of claws, and bare fingertips with fingerprints. Okay, then we're looking at 2 mammary glands, right? That's in green. And then the eyes with color vision, because we have cones. So we have color vision. So if you look at the slide, I've color coded it according to what they could ask you in the exam with regards to the head region, with regards to the arms, and with regards to the fingers, right? And the last one is sexual dimorphism. That is, we have separate male and females. There's a male organism and a female organism separately. So if you look at this diagram and you remember this, guys, it's not difficult to study this diagram. Every year you are asked this diagram, but make sure you know exactly which part you are being asked. And you can sit in the exam and look at your invigilator walking around and say, okay, head, freely rotating arm, eyes. I mean, you can get the answers from looking at the person next to you or your invigilator. So remember, when we see this little man, this diagram of this little man, we are speaking about the similarities, okay? So the similarities, this little guy and what everything is here. And look at the codes with colors. And or remember that we could ask you this different parts, right? Head region, arms, hands. So then you're going to speak about that particular part in the exam. Okay, I hope you're happy with that. I'm going to move on. Right, now that was the little man, that was similarities. Now we are looking at differences between humans and the African apes. Right, so if we look at this, the first one is bipedalism and quadrupedalism, right? The African apes walk on four, bi is two, bipedalism. Now, under bipedalism, we have 3 points which you need to know, and I am going to show you this with diagrams just now, right? Because it says study the differences with diagrams, and your exam guideline says that you have to study the differences and the similarities using diagrams. So we are going to use diagrams, and I'm going to explain this further to you. So if you look at the first one, the bipedalism, I said, right? Number one, the foramen magnum is in a forward position, and this is the key word, forward position. The S-shaped spine, the shorter and wider pelvic girdle compared to the quadruped organism, where the foramen magnum is towards the back, and it's a C-shaped spine and longer and narrower pelvic girdle. Cranium size, it's larger here and smaller here. Now remember, when we speak about cranium, the cranium houses the brain. So you're going to speak about the cranium and not the skull, because the skull is this whole area here forms the skull, but the cranium is the area that houses the brain. Then we're going to look at the palate shape, right? Semicircular or curved with regards to us humans, and rectangular or U-shaped with regards to the African apes. Then we're going to look at dentition. There's no gaps between the teeth and smaller canines with regards to humans, and the African apes, there are gaps between the teeth called diastema, and they have larger canines. Then we come to prognathus, that's a protruding jawline, right, or face here. Now, we're not using more prognathus and less prognathus anymore. We are saying non-prognathus and prognathus. So remember, we are humans, we have a flat face, right? So we are non-prognathus, and the African apes have a slope face, which I am going to show you. And then cranial and brow ridges are absent for us, but present for the apes. So this now is the differences that you need to know. Okay. Right, now we're going to discuss each of those differences. Now, the first one is bipedalism. If you asked about bipedalism, there are 3 points I told you that you need to remember. The first one we spoke about is the position of the foramen magnum. Now, if you look at these diagrams here, the one in a more forward position is the humans, which allows the spine to be vertical, and that is this one here, number 4. So this one is if this vertebral column is attached to that point, that is going to be a vertical rod, and that person is going to be upright. So bipedal. As you look at the trend going towards the more later years, you can see that as this foramen magnum moved from the point here where it was in number 1, where it was right at the back, it moves slowly forward to where we have it now. So the more backward position here is the African ape, and this one, if you have a vertebral column coming from there, it's going to be more of a horizontal column, and that organism will be walking on 4s. So remember then, bipedalism, the position of the foramen magnum, that is A, and now we're going to look at point B. Right, there's just some diagrams here to show you that this is in a forward position, and this is a vertical vertebral column, and this one is at a back position, and that is more of a horizontal position in which the spinal cord attaches to the foramen magnum, to the brain through the foramen magnum. Okay, let's see what we have next. The next one is B. Right, we're still under bipedalism, but we are B, and this is the spine. If you look at the spine for the ape, right, you can see it's a C-shaped spine here. It's curved. It's curved, and that will show you that the organism was bent forward, and quadruped, meaning walking on four. And if you look at this one here, it's more of an S-shaped or S-curved spine, and that is to support the lower body and the upper body, and that gives you the bipedal organism. Right, and now we're looking at C, under bipedalism, right? We A, B, and now we are on C, the pelvic girdle. The ape or the gorilla here has a long and narrow pelvic girdle. So if you look here from this end to this end, right, and it is short or small, narrow here, right? It's narrow here, but it is long, okay? It's long and it is narrow. And then if we compare this to the human, to us, you can see that it is much shorter, it is much longer across, right? So let's say it is short and it is wider. The word I was looking for is wider. It's wider across, but it is much shorter. Look at this arrow here is shorter compared to the longer arrow on this side. And this arrow here is much smaller or compared to narrower compared to the wide one on this side here for humans. Okay, so this is what you need to know when you're speaking about bipedalism and the differences between humans and the apes. Right, there's a diagram showing you the short and wide pelvic girdle here of the human. So it is, right, it is short and it is much wider compared to the narrowness here and the length there. Right, so this is just another diagram to reiterate what I was saying, and you can see here, right, that this pelvic girdle here is short and wide, and that supports an upright position, whereas if it is narrow here and long, as it is here in this part, then that supports a quadruped organism or on fours, right? And there's another diagram there to show you it is wide and short, and this one shows you it is long and narrow. So there's similar diagrams, but just to drill and make you understand that if it is bipedal, it is short and it is wide to support the upper body, and if it is narrow and long, then it supports the quadruped organism. Difference, and that is the size of the brain. Now remember I said the cranium is houses, or the brain sits in the cranium for protection, right? We have the cranium. So the cranium size is the next difference. And that would then indicate the brain size, because if it is a large cranium, obviously if the brain is inside, it's going to be a large brain. And if it is a small cranium, it's going to be a small brain. So the cranium is part of the skull where the brain is found. And a bigger cranium indicates a bigger brain size. And humans have a larger brain compared to our body size, right? And that would show that would indicate intelligence. A larger brain, intelligent. Okay, we now move on to the palate shape. So here we have the human with a curved smaller jaw here, right, curved, and the chimp with a more rectangular bigger jaw, or a U-shaped jaw. So here you can see again, right, we have the chimpanzee with a more of a U-shaped structure or rectangular structure here and here. And here, this is more curved. Can you see that? Right? So this is more U or rectangular, and this one is more curved. So this is more curved, and humans have a more curved palate. Okay? So now we're going to move on to the fourth one with regarding to the mouth, and that one is going to be dentition. Right. So if you look at the gorilla, okay, you can see here that they have large canines. Okay, there's a large canine there, and there's the canine here. So in between the canine and the incisor there is a large gap. There are spaces here between the teeth, and that gap between the teeth is called the diastema. And if you look at humans, we don't have a gap large gaps between our teeth. So we do not have a diastema. Okay, let me just go back there, sorry. Right, so we do not have diastema and they have, right? So when you are speaking about dentition, then these are the differences that you're going to talk about. And there's it here, you can see here. Okay, we don't have these spaces as you can see that they have in this diagram. Okay, I'm now going to move on to the next one, and that is number 5, prognathous, right? So as I said earlier, you are either prognathous or you are non-prognathous. And which, with regards to the ape, you can see this slanting, this position here, how this bottom end, the jaw, how it is protruding and coming outwards. So that means it is prognathous. It means having a projecting lower jaw. So African apes have a bigger jaw. Humans have a more flatter face than the African apes. And their face is more sloping. Look at the slope here, right? Look at the slope here. It's the same color as the line I'm drawing. And the humans have a chin. So there's no chin here. You can see this large area here. And this part here, we have a chin. Okay, so we are non-prognathous. The next one looks at cranial ridges. Okay, so if you look at humans, right, there are no huge cranial ridges. There may be these structures called sutures, when you were a young child and the brain was not fused yet to allow it to grow, and then it fused, but it did not form a ridge. It's just called sutures. Those are not ridges. The cranial ridges here are distinct, huge ridges. You can see these here, right? These are found in apes that are not found in humans. Right, so that's the cranial ridges. You can see here, there's one, and there's another one. And with humans, there's nothing. We don't have the cranial ridge. Right, number 7, we're going to move on to brow ridges, right? You can see here, there's a distinct brow ridge, and you can see it here as well. Okay, that's for the gorilla. And for humans, look at this. It is flat. There's no brow ridge, right? There's no brow ridge here compared to what we're seeing here with the apes. So remember, these are the differences. Now, when we look at the diagnostic report for 2020, this is an important document that your teacher will definitely highlight to you because these are the mistakes or the high frequency errors that were found in the NSC exams, right? It's called the diagnostic report, and this was for 2020. So what did learners say when they wrote the NSC exams for 2020? They said there's a difference between the skull. There is a difference between the skull and the cranium. But the learners spoke about a skull instead of identifying the word cranium. Now, terminology in life sciences is very, very critical, okay? So the cranium is part of the skull where the brain is housed. So if you are speaking about the size of the cranium, the size of the skull, it'll be marked wrong. You have to mention size of the cranium, not the skull. Okay, so you talk about the cranium, that's where the brain is housed, and not the skull, because the skull is the entire jaw, nasal area, eye sockets, everything makes up the skull. So you speak about the cranium. When talking about the differences between jaws of humans and African apes, as I said earlier, we don't refer to less prognathous, but we say prognathous and non-prognathous. We don't say less and more. Like previously, we use the word prognathous and non-prognathous. Okay, the next high frequency error that learners were making was the foramen magnum. In humans, you have to say it is in a more forward position, end of story. More forward position, that's it. No other word. So just learn it's in a more forward position. And then we find that learners are saying bipedalism is the ability to walk on 2 limbs, but that is incorrect because what are these? These are also limbs, but these are upper limbs. So bipedalism is the ability to walk on 2 lower limbs. That's the key there, not only limbs, because arms are also limbs. So you have to specify lower limbs or hind limbs, meaning limbs behind. That's what hind limbs are. Okay. Now, the diagnostic report for 2021. Candidates fared poorly in this question when asked to identify fossils found in South Africa. Right? This performance was unexpected since fossil names are clearly listed in the exam guidelines. So this section is actually more for next week when we speak more about the fossils, but I'm just going to explain to you now that you need to know the fossils found in South Africa. Guys, these are ours. It's our heritage. It's found on our land, but unfortunately, our learners don't know about what is on our land, and people all over the world know there's Sterkfontein Caves, and Littlefoot was found in Sterkfontein Caves. Taung child is found in Taung in the Northwest Province, but we are getting it wrong. So make sure you know your fossils found in South Africa, right? Because that is very, very important for us and for you to know about this and for the exams. Okay. Right, I'm now going to move on to some activities. So let's see. When you look at the activity, you're going to look at the diagrams and you're going to unpack the diagrams. So let's see, right? You're looking at diagram A. Let's see, that's diagram A. And the stem of this question will probably say, refer to the diagram below and answer the questions that follow. So let's look at the diagram. Right, we have diagram A. You can see it's a smaller skull region, a longer rectangular shape of the palate, and here it's a smaller palate. And then we're looking at X, that is a dot, but it is actually a hole in the skull region, in the skull. That one is more in the back. This one is in a more forward position. This one is a more curved palate. This one is a more rectangular palate. So immediately by looking at these diagrams, you can already see what the questions are going to be or what you may be asked, or you already know which skull is which here. I hope you are doing this with me and guessing it correctly, right? Let's look at diagram C, right? We can see here there's a more U-shaped structure there. There's a diastema here. Okay, there's nothing there, no diastema here, and this is a more curved palate there. Right, so this says provide a label for X, right? So what could X be? There it was at the back position. It moved now to a more forward position. It is a hole in the skull, and that is where the vertebral column will attach, and that must be the foramen magnum. Good, I hope you got that one. 1 mark there. The next question says tabulate 2 anatomical differences. Right. Firstly, the instruction is tabulate. When you see the word tabulate, immediately it means draw a table. If you don't know anything else and you draw a table, you'll get 1 mark, right? So immediately T for tabulate, T for table. The second thing is 2, and the 2 is specified. Therefore, we'll mark the first 2, and you need to give the 2 most important points. And then it says anatomical differences. Anatomical meaning structural differences, anatomy, right? Between the jaws in diagram C and D, which represent trends in human evolution. Now, guys, be careful here. The other key word here is jaws. So we do not talk about differences and speak about the other differences like the arms. We have freely rotating arms. Okay, that one is similarities. Here's a difference now. But you can see the foramen magnum here is in the back position, and here it is in a more forward position. So we're not talking about those kind of differences. The difference that is stated in the question is saying jaws. So keep to the question. Take a highlighter with you in your exam. Highlight these key words. So now that means we are not looking at diagram A and B. We are now looking at diagram C and D, right? So tabulate 2 anatomical differences between the jaws of diagram C, in diagram C and D, which represent trends in human evolution, right? So we'll talk about that now. Let's just answer the question. So there's your table. The table, you get 1 mark for the table. There's your diagram C and your diagram D. Right, let's look at it. for diagram C, there are large canines. There's your large canines there, and there are smaller canines here. Now, this is important when you are filling in a table. When you are asked to tabulate, make sure that what you're speaking about as your first point on the left-hand side, you carry through the same point on the right-hand side, or you give the difference on the right-hand side, so that you are comparing. And that's what we want to see. We want the difference. You want to see that you are comparing. So if you're speaking about canines, then that whole row must be about canines. So if you are saying there are large canines for diagram C, then you need to speak about canines for diagram D. And with regards to diagram D, there are small canines there. Right. Then the next difference is you can see the spaces here, the diastema. So there are spaces present between the teeth, or there is diastema between the teeth for diagram C. And for diagram D, there's no diastema or no spaces between the teeth. Number 3, if you look at, they asked for 2, right? But we are just giving 3 here now. And obviously, you put the 2 most important ones at the top, and the examiner will mark the first 2. But I'm just giving you an option of what there is that you could give as an answer. Here, the jaw is U-shaped, okay? And then the jaw here is C-shaped, or this one could be rectangular, and this one could be more curved, right? So can you see that? Guys, look at these questions and see what you could be asked. Let's look at it's the same activity, just another question. Give the letter only, okay? Give the letter only of the diagram that represents the skull with a larger brain capacity. So if that skull is going to have a larger brain capacity, it's going to be one that has the skull with the larger cranium size. And if you look here, you can see here, right, that this is a smaller area and this is a much bigger area here. So I'm going to say that that one is B. And that is correct. The next one says, account for the position of X in the skull in diagram B. So we want you to account for this position X, right? So we've already established in the first few questions that X is the foramen magnum. So now they want you to say, account for the position. That means, why is the foramen magnum in this position that it is? So if we look at skull B, we can see that the position is in a more forward position. Remember, I said, remember the word forward. It's in a more forward position, and therefore it is going to be the foramen magnum, or X, is located below and central in a more forward position in the skull. So let's just take out all of that. Let's say in a more forward position in the skull. And then one more question I left out, which is a more prognathous skull, and that then will be A. That's going to 1.3b here. A more prognathous skull, right? Which one is a more prognathous skull? And if you look at this, you can see here the dentition, this is jutting out compared to this one here, right? So this one here, diagram A, would be the one with a more prognathous skull. And then we said here the foramen magnum is located in a forward position, right? Below, in a more forward position in the skull. Okay, and you'll get your mark for the foramen magnum and in a forward position over there. Right, so the next activity, also, let's just see, sorry, the organism. This shows us that the organism is bipedal since the spinal cord enters vertically, right? The spinal cord is going to enter vertically there. So this organism must be bipedal, and that must be the human, which is organism B, with diagram D as the palate. Right, 1.5 says, explain the significance of the shape of the spine that is associated with skull B with the skull in diagram B. Now, we've established that this must be the human. So obviously, if we are looking at this foramen magnum in a more forward position and the vertebral column is going to be vertical, then that spine is going to be an S-shaped spine. So the spine is S-shaped, And the reason for that, it says explain the significance. That means it wants you to tell us why, right? Why the shape is what it is. So the shape is S-shaped. You get a mark for identifying that organism B is a human and therefore it has an S-shaped spine. And the significance of it is to balance the weight above the hips or to support the weight of a bipedal organism. Right, the next activity is name the term used to describe the type of locomotion of species B. Let's look at that locomotion and we can see that there's 2 there and 2 down here. This is 4, therefore this must be a quadrupedal organism, right? Quad 4. Explain how the position of the foramen magnum in number 2.2, the shape of the spine, and the structure of the pelvis are suited for the type of locomotion in species A. So this is a fully loaded question for 6 marks. Firstly, it's an explain question. Right? Number 1, it's an explain question. That means you need to give the structure and the function, a cause and an effect. How the position of the foramen magnum. Number 1, you're going to speak about the position, the foramen magnum, the shape of the spine, 2, the shape of the spine, and the structure of the pelvis. The structure of the pelvis is number 3, are suited for the type of locomotion in species A. So look at the diagram. I'm going to grab a sip of water while you're looking at that. Right, excuse me. Let's move on. So you can see here, explain how the position, the shape, and the structure of the pelvis. So that is why your highlighter is so important, because you are going to explain all these 3 parts, and it's 6 marks, and you're only asked to explain 3 foramen magnum shape and structure of the pelvis. So that means you have to give a cause and an effect, right? There has to be another part to your answer that substantiates what you are saying in the first part. So let's look at the answer. The foramen magnum is in a forward position, right? The position of the foramen magnum, it's in a forward position, right? We're looking at in species A. So this here is species A, right? That organism. The foramen magnum must be in a forward position, 1 mark, to allow the spinal cord to enter vertically, right? That's the 2nd mark. Then we look at the structure of the pelvis. The structure of the pelvis. Okay, there's it here, the last point. The pelvis is short and wide, or you could say broad, to support the upper body in an upright position or bipedal position. And then we move to the second part of the question that says the shape of the spine. The spine is more curved or S-shaped to absorb shock and to allow for flexibility and for movement or for support. However you explain it, but it's any one of those that you could use to explain your answer. I hope you understood all of those and got all those right. Let's look at another one. Right, this is differences between humans and African apes. The diagram below shows 3 skulls, right? 1, arrange the skulls from oldest to youngest. Youngest meaning more recent. So we're going to look at the cranium size. Look at that size of that cranium. Look at the jaw. There's a chin there. Look at this here, right? The cranium size is a bit smaller and not much of a chin there. And we have a cranium size there. And look at this, okay? So we're going to see that the more, arrange them from the oldest to the most recent. Okay, so we're going to say that the oldest one, the youngest one, the most recent one is going to be A, because this one looks more like a Homo sapien, right? So our answer is going to be B, which is the oldest, right? More broader here, smaller brain, that's going to be 1, that's going to be 2, and that's going to be 3. So your answer is B, C, A. 2 marks or nothing. Right, next one. Describe the significance of the difference in cranium size between skulls A and C. Which ones are we talking about? We're speaking about skulls A. Put a star so you know which one you're looking at in the exam. skull A and skull C, and the significance in the cranium sizes between those skulls. And if you look at skull A, it is larger than skull C, right? The cranium size of skull A is larger than the cranium size of skull C, okay? Because they talk about the difference in cranium size. So remember, you're speaking about cranium size and not skull size. So the difference is the cranium of the skull A is larger than the cranium of skull C, indicating an increase in brain capacity, and that means more intelligent. Okay, describe two visible differences, visible, what I can see, and two of them in the structure of the jaw of skull A and B, the jaw of skull A and the jaw of skull B. Right, so skull A is not prognathous or less protruding than skull B, and skull A has a well-defined chin, whereas skull B does not. So there you got your 4 marks there for that one. Right? You could also say skull A has a smaller jaw, whereas skull B here has a much larger jaw. Okay, I'm going to look at just two more slides. Write down the letters only of the diagrams that represent bipedal organisms. So the letters only, if you are asked for letters only, make sure you give letters only, right? Of the diagrams A to D that represent bipedal organisms. So let's look at this. Right, here we have a long pelvic area, and it seems to be a bit narrow. This one here is much wider and it is shorter. Okay, so we are looking at this diagram here, right? Bipedal, it must be this one here. And then if you're looking at the foramen magnum in a more forward position, look at the dentition. That will give you a clue. We're looking at those two, A and D. Explain how the shape of the pelvis contributes to bipedalism. The shape of the pelvis. Here we have a long, narrow pelvis. Here we have a wider, shorter one in this diagram. Okay, so the pelvis is wide, right? How does it contribute to bipedalism? That means how does it make bipedalism easy, or why is it present there for bipedalism? The pelvis is wide to support the weight of an upright organism or a bipedal organism. So wide pelvis supports the weight of an upright organism. Right, and then explain the significance of the changes in teeth of humans, which shows progression in evolution. Remember, by us learning about the apes and learning the progression, the different skulls, and what is the differences between humans and apes, it shows us evolution. It shows us progression. It shows us how we evolving, becoming more modern, becoming more modern, right? Okay, so let's look at this. Explain the significance of the changes in the teeth of humans, which shows progression in evolution. Now, if you look at the teeth here, the large canines, it means that they were pulling raw meat, hard meat, right? And look at our dentition is much smaller. So our diet is different to what their diet is. And your dentition can tell you a lot. When we find the fossils, by looking at the teeth of all these organisms, dinosaurs, whatever organism it is, we can tell you what that organism ate because of the size of the teeth and how it is, right? So if you look at our teeth, it is smaller, which means we're not eating raw meat and ripping meat and pulling it apart, okay? So for 4 marks, canines have changed from large to small, okay? So that's the evolution. From large, it went to small, right? And this is due to the change from eating raw food to eating cooked food. Our food is more tender because we cook it, and therefore it's easy to chew. It's easier to chew our food because we have, that is why we have smaller teeth and smaller canines. Right, guys. Thank you very much for your time. My name is Fawzi Rahman, and I will be with you next week again with Sina, and we will be doing the next part or the final part of evolution. So make sure you come and you be with us again next week. But thank you to my colleague Vedika for an awesome presentation as well. And we hope that you go over everything that was taught to you and prepare for this important section, which is 54 marks in the exams, 1/3 of the paper, and you can score on this section alone. You can pass your exams. So all the best to you guys, and take care, and see you back again soon. Goodbye.